Hello, and welcome to part 2 of today's lab. In part 1, we looked at histology. In part 2, we're looking at models. It's a very short lab today. Normally, I take students into the cadaver lab and actually start going over muscles, but since we have several labs for the muscles, I'm just giving you a short lab today. So, this is skeletal muscle. In skeletal muscle, you have long, parallel cells, and it's striated. So remember, light, dark, light, dark. And we're going to look at better pictures of this. It has multiple peripheral nuclei. And then we have this junction between the axon, which is from the nervous system, and the muscle. So this, from the central nervous system, you have something called an axon. We're going to learn it sends messages. And the axon splits into these branches called telodendria. That means faraway branch. And it has to go to every single muscle fiber. So we're going to look at this model in a couple seconds of a close-up of this junction between the axon bulb and the muscle. However, since I'm on this, I'll go over the other two muscle types. So this is cardiac muscle. Again, striated, light, dark, light, dark. Central located nucleuses and the intercalated discs. Then for the smooth muscle, we have the tapered cells, but which is called fusiform. Instead of one axon with little branches called telodendria going to fibers, we have these swellings. And I have a love-hate relationship with these models. I love to hate them. What should be shown is, yeah, it goes here and here, then there's another viscosity there, then another one there. So you just have these swellings throughout. So they didn't do a good job of differentiating between the axon bulb and the varicosity. So this is a single muscle fiber and all the motor neuron. This right here is the sarcolemma. That is the cell membrane. Then over here, we have the endomysium that's surrounding it. Now, if we zoom in, we can see the myofibrils. This is the Z-disc right here. Don't worry, we'll get a better sarcomere. That's the Z-disc right there. We have the M line right here. There's the M line. We have the myosin right there. We have the actin right there. So this right here would be the I band. And then this right here would be the A band. So that's the inside of the skeletal muscle. But we care about the motor end. So this structure right here is called an axon. And an axon takes messages from the nerve cell body, and it takes it to a effector or end location. Now at the end here, it's branching into these bulbs. These are called synaptic bulbs. So those are synaptic bulbs right there. And if you notice, the synaptic bulb actually is not touching it. There's a gap there called a synapse. So this is the synapse. And then this is the end plate right there. So I'm going to have you look at it. You're going to try to identify the parts of the striations. You're going to identify the axon bulbs, the axon. And I've gone back and forth between teaching the axon first and then teaching this first. I have not found a happy medium. So I want you to draw a sarcomere. You can draw this one. Now what I want you to do is you are going to label the parts of a sarcomere. And I drew, I think, 20 of them in lecture. But just to go over the parts, oops, let's make this a little bit bigger. We have an M line right there. M line, middle line. And then we have myosin. So this is the myosin. I swear I picked a different color than that. I guess not. And has these golf club-like heads that want to grab on to this thin actin right here. So this is the actin right here. And the actin goes to this Z-disc over here at the end. So see how it looks like a Z? That's the Z-disc. Now this zone right here, where I only have myosin, is the H zone. Now, there's also an I band and an A band. The A band goes from the beginning of the myosin all the way to this side of the myosin. Now, the I band, this right here, where there's only actin to the Z disc, is half an I band. So the I band would start here, go over, continue on to the next sarcomere, and go here. Quick 30-second photoshopping. The I band would start right here. It would go to this side. So the I band really crosses the Z disc. So the A band is from the beginning of the myosin to the other end of the myosin. The I band is from here, where the myosin ends, to where it begins on the next sarcomere. So I want you to draw it. Now I want to zoom in a little bit more, because you're going to be looking at this online. I want you to notice the actin here. The actin has two other molecules on it. This molecule right here is called tropomyosin. Now, the tropomyosin is blocking these myosin heads from grabbing onto the active site of the actin. There's this little Mickey Mouse molecule right here that is called troponin. This is the key. So calcium will bind to the troponin, moves the tropomyosin away from the active site, myosin will grab it. So when you go to the website, I'll ask you those structures as well. So here's a different picture. So on this picture, the A band would start there and go that way, crossing it to the other side. So the A band really is where you have exposed filaments. So I really want to show you a different version of this picture that wasn't labeled, but I couldn't find it. This is an art project that somebody did that shows skeletal muscle. So here we have the actin and the myosin. Again, the I band is from one side to the next side. Z disc is in the middle. The A band is from beginning of myosin to the end of myosin. M line's the middle. H zone is where you only have it. Now this is also showing mitochondria in here, the peripheral nucleus. And this network is the sarcoplasmic reticulum. So I might actually show you a slightly different model that combines this one and the first model as well on the lab exam. So look at all three, and thank you. That is all for the models. Have a good day.